I'm Dr. Anjani Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. As you pretty well know that I'm dealing with the course Enzyme Technology, and we are presently dealing with the Unit 3 of your Enzyme Technology. In the last sessions, we have dealt with the kinetics of single substrate reactions. We have already covered the estimation of Michaelis-Menten's parameters. Along with that, we have also learnt about what is the importance of your Michaelis-Menten parameters like Km and Vmax. Along with that, we have already studied about multi-substrate reaction mechanisms and kinetics, and we have focused upon the turnover number also. In the present sessions, we are going to study about the remaining topics of your Unit 3. We'll start with competitive inhibition. What is this competitive inhibition? As the name indicates that the inhibitor is competing with the substrate molecules to bind with the active site of the enzyme. Here, the inhibitor targets the active site of the enzyme. Actually, substrate goes and binds to the active site of the enzyme. Now, your inhibitor is also targeting the active site of the enzyme, because of which there is a competition between your inhibitor and the substrate for the active site of the enzyme. So, who will win in this particular competition? The compound whose concentration is high definitely wins the competition. Let me explain this in depth. Competitive inhibitors compete with the substrates for the active site of the enzyme. Moreover, a competitive inhibitor typically has close structural similarities with your substrate molecules, because of which it is having compatibility to bind with the active site of the enzyme. So, your inhibitor is having the structural compatibility with your substrate molecules. So, we have to note this point. So, your inhibitor is having your close structural compatibility with the substrate molecule. Thus, it competes with the substrate molecules in order to bind to the active site of the enzyme. So, here in this particular situation, the enzyme may bind either to substrate molecule or to an inhibitor molecule, but not to the both of them. We have to remember this. So, enzyme is going to bind either with the inhibitor or with the substrate molecule, but not with both. So, binding to the inhibitor inhibits the activity of the particular enzyme. Moreover, the competitive inhibitors bind reversibly to the active site of the enzyme. We are studying the competitive inhibition under the category reversible inhibition. That means your competitive inhibitors bind reversibly to the active site of the enzyme. So, the enzyme bound inhibitors either lack appropriate reactive groups or held in an unsuitable position with respect to the catalytic site of the enzyme. How the inhibitor is going to bind with the enzyme? The inhibitor which is going to bind with the enzyme is either lacking an appropriate reactive site or it is not properly held in a suitable position with respect to the catalytic site of the enzyme. You just see here, as your enzyme is present, actually it should bind with your substrate molecule to give rise to your enzyme substrate complex. But your inhibitor is going and binding with the active site of an enzyme in order to form enzyme inhibitor complex. So, once this enzyme inhibitor complex is formed, we are referring this complex as dead end complex. Why we are calling it as a dead end complex means there is no probability of formation of product. That's why we are calling this enzyme inhibitor complex as dead end complex. But I've told you that the situation can be reverted when in the presence of high concentration of your substrate. So, at high substrate concentration, the action of this competitive inhibitors can be overcome because of sufficiently high substrate concentration successfully competes out with the inhibitor. As your substrate molecules are more in number, they will be competing with your inhibitor molecules for the active site of the enzyme. So they won't allow, or if at all inhibitor binds also, they will drag the inhibitor out and they will be binding to the active site of the enzyme. So because of this competition between your substrate and the inhibitor in the presence of high concentration of your substrate, there is every chance that you can revert back the situation. So many drugs work by mimicking the structure of your substrate molecule, which will be targeting a specific enzyme, and hence we act as competitive inhibitors of the enzyme. I'll explain you with the help of a figure. Just see this. This is your enzyme molecule. The enzyme molecule is having a proper active site. Along with the active site, enzymes will also have other sites which we are calling as allosteric sites. Okay, so enzyme will be definitely having an active site which is very specific to your substrate molecule, but the enzymes will also contain other sites which are called as allosteric sites. So substrate molecule should go and bind to the active site of the enzyme. So if at all in place of substrate molecule, your inhibitor is going and binding to the active site of the enzyme, here there is a formation of enzyme inhibitor complex in place of enzyme substrate complex. And because of formation of this enzyme inhibitor complex, you are going to get a dead end complex because you are not going to get any sort of product here. So inhibitor here binds with the active site of your enzyme and no more reaction is going to take place. This inhibitor is blocking the active site of the enzyme. I'll explain you the concept with a suitable example. Let us consider an enzyme which is nothing but your succinate dehydrogenase. We are considering an enzyme which is nothing but your succinate dehydrogenase. So what is the reaction which is catalyzed by this particular enzyme? So succinate is getting converted into fumarate in the presence of succinate dehydrogenase. So the reaction catalyzed by this succinate dehydrogenase is conversion of succinate to fumarate. From the figure you can see that the succinate is having two carboxyl groups. Along with that there are two more carbon groups. If you see the fumarate, it also contains two carboxyl groups and a double bond between your two carbon groups. Here malonate is acting as a inhibitor to the succinate dehydrogenase. Let me explain about the mechanism here. If you see malonate here, malonate is also having two carboxylic groups, but it is containing only one carbon group here. There is every probability that this malonate can come and bind to the active site of an enzyme succinate dehydrogenase in place of succinate. Actually, succinate should go and bind to the active site of succinate dehydrogenase. But this malonate, what it is doing is, it can go and bind to the active site of an enzyme. So, pretty well, as it is having some structural similarity with the axial substrate, it is going, your malonate is going and binding to your succinate dehydrogenase. But there won't be any formation of product. This malonate cannot involve in the formation of your fumarate. Why? Because if you see the malonate structure, you have only one carbon group here. But in the case of succinate and fumarate, there are two carbon groups. This malonate is not involved in, it can't go for the generation of a double bond between the two carbon groups present in fumarate. So, even though malate is having the structural similarity with the succinate, It can go and bind to the succinate dehydrogenase active site, but it can't go for the production of your fumarate because of lack of carbon groups in the center. It is in deficit of one carbon group and it can't form the double bond between the two carbon groups. And what is the effect of your competitive inhibitors? And what are the various components on which the competitive inhibitors effects depends upon? So, the effect of competitive inhibitors depends upon the first thing, inhibitor concentration, substrate concentration, relative affinities of your substrate and your inhibitor. Let us consider the same concentration of your enzyme and your inhibitors and less substrate concentration. I am just giving the condition here. Let us consider equal concentration of inhibitor and enzyme with less concentration of your substrate. What will happen here? The degree of inhibition will be high because the concentration of your substrate is low. Definitely the degree of inhibition will be high. Let us consider the second situation where the concentration of your inhibitor and enzyme are same, and now we have considered excess amount of your substrate. Now what is the situation? Definitely the degree of inhibition will be low because the inhibitor's concentration is comparatively lower than your substrate concentration and the effect of inhibitor will be definitely low. It will be reduced in presence of high concentration of your substrate molecule. Let me explain you the effect of Km here. See, while apparent Km is affected by the addition of the inhibitor, the value of Vmax does not change. During this competitive inhibition process, there won't be any change in the Vmax, but there will be increase in the value of Km, which we are representing as apparent Km. So, apparent Km will be increased by the value which is equal to 1 plus the concentration of inhibitor divided by Ki. I'll explain you how we have got that particular expression. Here, this figure show you the clear idea of what is happening in inhibition process. There is enzyme, substrate is actually coming and binding to the enzyme in order to form the enzyme substrate complex, which will be resulting in the formation of enzyme product. But in presence of inhibitor, this inhibitor is going and binding to the enzyme in order to give your enzyme inhibitor complex, because of which there won't be any formation of your product. Moreover, if you see this particular plot, in presence of your substrate concentration versus your rate of reaction, in absence of your inhibitor, the curve will be in the hyperbolic shape. But in the presence of competitive inhibitor, we are seeing a sigmoidal shape curve. For this competitive inhibition process, what is the expression of Michaelis-Menten? It's nothing but V is equal to Vmax into substrate concentration divided by Km into 1 plus concentration of inhibitor divided by your Ki plus the concentration of your substrate. Here, what is Ki? Ki is nothing but your axial enzyme inhibitor complex dissociation constant. It is a constant related to your enzyme inhibitor complex. So, we are calling it as dissociation constant. We just see this slide in order to know how we have got that particular expression which I have shown you in the previous slide. As we pretty well know that the enzyme substrate reaction is a reversible reaction. You can see both forward as well as the reverse reaction. You can get this enzyme substrate complex which will be leading towards the formation of enzyme and product. But in presence of your inhibitor, you are getting this enzyme inhibitor complex. Here, if you see what is Km, Km is equal to the concentration of your enzyme into concentration of your substrate divided by the concentration of enzyme substrate complex. Moreover, what is Ki? Similarly, the dissociation constant for this inhibition, there is nothing but your competitive inhibitor, is equal to the concentration of enzyme into concentration of inhibitor divided by the concentration of enzyme inhibitor complex. So we pretty well know that until now, what is the total concentration of your enzyme? So your enzyme, the total enzyme may be present as some amount of enzyme may be present in its free form, some amount of enzyme may be present in its bound form. So in order to calculate the total enzyme, you can add your free form of enzyme along with the bound form of enzyme. But in the presence of inhibitors, in order to calculate the total concentration of enzyme, you have to include another expression which is nothing but E total, which we can also denote as E naught. Your E naught is equal to your free enzyme plus the bound enzyme plus the enzyme which was bound to your inhibitor. So E naught is equal to E plus ES plus EI, and E naught is equal to E plus ES plus, in place of EI just incorporate the expression E into concentration of inhibitor divided by Ki. So by doing all the substitutions and rearrangements, we can get the final expression as V is equal to V max into S divided by S plus Km into 1 plus I by Ki. So if you see this until here, it's nothing but your Michaelis-Menten equation. Here the Km is getting increased by this particular fraction, is nothing but 1 plus concentration of inhibitor divided by Ki. So if we define your Km, its apparent Km is equal to Km into 1 plus concentration of I divided by Ki. So if you consider, this is your Michaelis-Menten equation. Let us consider the Line-Weaver-Buck equation, which we will be obtaining by taking the inverse of your Michaelis-Menten equation. If you take the inverse, we will be getting the expression like this. 1 by V naught is equal to Km by V max into concentration plus 1 by V max. This is nothing but your Line-Weaver-Buck equation. Let me explain the second type of your reverse inhibition process, that is nothing but your uncompetitive inhibition process. So what's happening here? It's a sort of a reversible inhibition process, as we pretty well know that uncompetitive inhibition is nothing but your reversible inhibition process that occurs when your inhibitor only binds to the enzyme substrate complex and not to the free enzyme. Here you have to know that your inhibitor is going and binding to the enzyme substrate complex. It's not going and binding to the free enzyme here. Let us see this particular figure. Consider this as your enzyme and this is your substrate molecule. When these two combines, you will be getting a enzyme substrate complex. In a normal situation, you will be going to get the enzyme product also. You are going to get the product with the release of your enzyme. But in the presence of inhibitor, let us consider this as inhibitor. This inhibitor I is going and binding to the enzyme substrate complex. It is not going and binding to the enzyme. It has more affinity and it will be going and binding to the enzyme substrate complex. That means, can you guess where this inhibitor is going and binding? As you know that the substrate is generally binding to the active site of enzyme, if at all your inhibitors should bind to the enzyme substrate complex, it should bind to the another allosteric site of your enzyme. So your inhibitor is going and binding to the allosteric site of your enzyme in order to form your enzyme substrate inhibitor complex. So it is nothing but your ESI complex. Here we are getting your enzyme substrate inhibitor complex, which is not allowing further reaction where you will be normally getting your product, but as you got enzyme substrate inhibition complex, a dead end is formed, a dead end complex is formed. This enzyme substrate inhibition complex is a dead end complex. So there won't be any probability of formation of your product. So one can hypothesize that on binding, yes, a conformational change will occur to the enzyme molecule, which presents a binding site for your inhibitor. So once your substrate is going and binding to the enzyme, now there will be some conformational changes that is taking place in your enzyme, and now your enzyme is exposing another site for the inhibitor to bind. That I am calling it as allosteric site. So inhibition occurs since ESI cannot form product. I have told you that ESI is dead end complex and we are not going to get any sort of product. So what is the fate? What is the situation of your Km and Vmax? Both Km and Vmax are altered. In the previous case, when you have studied about your competitive inhibition, there I have told you that there won't be any change in Vmax and there is increase in Km. Here, in this particular uncompetitive inhibition process, both of your Km and Vmax will be altered. Let us continue. In the presence of your inhibitor, as substrate increases to infinity, not all the enzyme is converted into enzyme substrate. That is, there is a finite amount of enzyme substrate inhibitor complex even at infinite S. So, you can see that by increasing the concentration of your substrate, you can get back your original situation. There won't be any impact of your increased substrate concentration on the uncompetitive inhibition. Your Vmax is equal to K cat into E naught. If and only if all the enzyme is in the form of ES, that is nothing but enzyme substrate complex formation. Under these conditions, the apparent Vm, which is nothing but your apparent Vmax, your Vmax is lesser than that of your Vm. In the last slide, I have told you that here there is a change in your Vmax as well as your Km. Here, the Vmax is told you that there is a change. What is the change here? Vmax is going to get decreased without inhibitor. In addition, the apparent Km, which I am representing as Km app, will also be changed. And we can use Lee-Chatelier's principle to understand this particular concept. If your inhibitor is going and binding to this enzyme substrate complex alone and not with your enzyme, it will shift the equilibrium of ES towards enzyme substrate to the right side, which would definitely have the effect of decreasing the Km. That is, it would appear that the affinity of enzyme and the substrate has decreased. So, what is the expression for this particular uncompetitive inhibition process? So, it's a lengthy expression. V naught is equal to Vm by 1 plus I by Ki into S divided by Km by 1 plus I by Ki plus S. So, from this concept we have understood that there will be difference, there will be change in your Km and Vmax values in the case of your uncompetitive inhibition process. Let us now deal with this non-competitive inhibition. What is this non-competitive inhibition? This non-competitive inhibitors bind to the enzyme. So, in the competitive inhibition process, I have told you that your inhibitor is going and binding to the active site of the enzyme. In the case of uncompetitive inhibitor, I have told you that your inhibitor is going and binding to the enzyme substrate complex in order to form ESI complex. But in the case of non-competitive inhibition process, your inhibitor is free to either go and bind with the enzyme in order to form EI complex, or it can also go and bind to the enzyme substrate complex in order to form ESI complex. So, non-competitive inhibitors bind to the enzyme or enzyme substrate complex at the site different from the active sites, decreasing the activity of the enzyme. A non-competitive inhibitor binds reversibly at the site other than the active site and causes a change in the overall three-dimensional shape of your enzyme that leads towards a decrease in the catalytic activity of your enzyme. So, what is happening? Your inhibitor is going and binding to the site other than your active site and bringing some changes in the catalytic activity of your enzyme. Since the inhibitor binds at a different site from the substrate, the enzyme may bind to the inhibitor, the substrate, or both the inhibitor and substrate together, as I have already told you. So, the effect of non-competitive inhibitor cannot be overcome by increasing the substrate concentration. So, there will be definite decrease in the Vmax value without any change in your Km value. So in the case of non-competitive inhibition, Vmax will be decreased and there won't be any change in the Km. And we can study the example of pepstatin, which will be affecting the enzyme renin as an example for your non-competitive inhibition. We can take the effect of pepstatin on the enzyme renin as an example for your non-competitive inhibition process. So, I'll explain you with the help of a figure. See here, I've told you that it's an enzyme having two sides. One is for your substrate and there's a probability of inhibitor can go and bind to the another side of your enzyme. So, this is your substrate molecule, this is your inhibitor. So, the substrate is going and binding to the active site of the enzyme, whereas your inhibitor is going and binding to the another site of your enzyme in order to form a enzyme inhibitor complex, which is nothing but your dead end complex. Here the inhibitor is binding to the allosteric site and it is going to bring changes in the active site of the enzyme. If you consider the competitive inhibition process, your inhibitor is going to bring changes in the active site, whereas in non-competitive inhibition process, your inhibitor is going to bring drastic, is going to bind. In the competitive inhibition process, your inhibitor is going and binding to the active site of the enzyme. But in the case of non-competitive inhibition process, your inhibitor is going and binding to the allosteric site of the enzyme. As an example for this particular process, I can discuss with you the effect of this particular heavy metal. That is nothing but heavy metal poisoning. So, I'll explain you. Certain compounds containing this heavy metal such as lead, mercury, copper, silver, etc. They are very much dangerous. They are very much poisonous. This is because of the ions of these metals are non-competitively inhibiting the enzymes. These metal ions are acting non-competitively. They are acting as non-competitive inhibitors with the enzymes. So, the silver ions react with the SH group that is present in the side group of cysteine residues in the protein chain. Let me show you what's happening here. Let us consider this as a protein containing this particular amino acid cysteine. It contains a sulfidyl group, SH group. Let us consider a metal component, Ag. This Ag is going and binding to the SH group of your cysteine residue of the protein chain. See here, Ag is going and binding to the S of your cysteine residue. There isn't enough electronegativity difference between the silver and sulfur. Here, there is no enough electronegativity difference between silver as well as the sulfur for a full ionic bond. That's why the bond can be only considered as covalent bond. Here, the bond that is formed is your covalent bond. If the cysteine residue is somewhere on the protein chain, which affects the way it folds. So, what is the effect it is showing? Once your metal is going and binding to the sulfur group of your cysteine, what's happening? It's affecting the folding of your protein into your tertiary structure, which is having effect on the active site of your enzyme. So, it will be stopping the enzyme from working. So, with the addition or binding of your metals with the sulfur group of your cysteine, they are hampering the folding of your protein molecule. Thereby, there will be change in the active site of your enzyme, which is completely stopping the effect or activity of your enzyme. Moreover, we will study about mixed inhibition now. From the heading itself, we can understand that it's a inhibition which is a combination of two or more or one or more inhibition process. We are now going to study about your mixed inhibition. From the name, we can analyze or we can understand that it's a mixture of several inhibition processes. So, coming to this mixed inhibition process is a type of enzyme inhibition process in which the inhibitor may bind to your enzyme whether or not the enzyme has already bound to your substrate molecule. That means it can either bind to the inhibitor that is involved in this particular inhibition process may go and bind to enzyme directly, or sometimes it may bind to the enzyme substrate complex also. So it is called as mixed inhibition because it is a conceptual mixture of competitive inhibition, where if at all you are considering it's a mixture of competitive inhibition, means according to competitive inhibition, your inhibitor should go and bind to the active site of the enzyme. So this mixed inhibition means it's a conceptual mixture of your competitive inhibition process and your uncompetitive inhibition process. So as like your competitive inhibition process, your inhibitor can go and bind to the active site of the enzyme, and as similar to your uncompetitive inhibition process, they may bind to the enzyme substrate complex. The inhibitor may go and bind to the enzyme substrate complex. So uncompetitive inhibition, in which the inhibitor can only bind to the enzyme if the substrate has already bound to the enzyme. If the ability of the inhibitor to bind to the enzyme is exactly the same, whether or not the enzyme has already been bound to the substrate, it is known as non-competitive inhibition. So we know what is the main differences between competitive, non-competitive and uncompetitive. Competitive inhibition process is that process where your inhibitor is going and binding to the active site of the enzyme. Uncompetitive inhibition process is that process where your inhibitor is going and binding to the enzyme substrate complex. It can go and bind to the enzyme alone. And now considering the non-competitive inhibition process, your inhibitor can go and bind to the enzyme, or it can also bind to the enzyme substrate complex. So a mixture of these inhibitors can be considered as mixed inhibition process. Non-competitive inhibition process is sometimes it is considered to be the special case of this particular mixed inhibition process. It's nothing but your non-competitive inhibition process. In mixed inhibition, the inhibitor binds to the allosteric site, as we have already discussed. That means it will bind to the site different from the active site of the enzyme, where normally your substrate will be binding. However, not all the inhibitors that binds to the allosteric sites are mixed inhibitors. Mixed inhibitors may result in a decrease in the apparent affinity of your enzyme for the substrate. That means your Km value appears to be increasing when your inhibitor favors binding to the free enzyme, more closely mimics the competitive binding. What I want to discuss with you is, due to this mixed inhibition process, you can get two sorts of results. The first one is, because of this mixed inhibition, there will be decrease in the apparent affinity between your enzyme and substrate. Affinity between this enzyme substrate may be decreased, which will be leading towards the increase in the Km value. I've told you while discussing your Michaelis-Menten equation is, if your value of Km is more, the affinity between enzyme and substrate will be less. So, because of this mixed inhibition, there may be a chance of decrease in affinity between your enzyme and substrate. Because there is decrease in affinity of this enzyme substrate, we can say that there is increased Km value when inhibitor favors the binding to the free enzyme. Another case is, another result may be an increase in the apparent affinity may take place between the enzyme and substrate. So, there might be increase in the affinity between enzyme and substrate. That means your Km value is less. Km appears to be decreased when the inhibitor favors binding to the enzyme substrate complex. More closely, this is mimicking the uncompetitive binding. So, in either of the cases, in either of the inhibition processes, there will be decrease in the enzyme reaction rate. Coming to the summary, until now in this session, we have discussed about what is enzyme inhibition, what are inhibitors, what are the various types of inhibition processes. In that we have discussed about your irreversible inhibition, we have discussed about your reversible inhibition. Under irreversible inhibition process, we have studied about your suicide inhibition process. Under reversible inhibition process, we have studied about competitive, non-competitive and uncompetitive inhibition processes. In that, we have also studied about how your Km and Pmax values changes from one inhibition process to another. I want you people to work out these questions so that you can assess yourself how much you have understood from today's class. Moreover, you can refer to these references. This is a textbook and this is a slide share. So you can refer these particular references in order to gain more. Thank you.